So I'd like to thank Dept and the organizers for the invitation. And if I'll know how to move this, will be great. So I'll be studying how to manipulate cellular function in order to generate novel therapeutic approach to treat mainly leukocytes-implicated diseases. This was very unappreciated about 20 years ago, and now it's highly appreciated. So it's good to be on the wave again. And we have a lots of things that we are working on, blood cancer, inflammation, viral infection, and for us, this is a great opportunity. And the lab is basically designed into two major groups: people that are doing drug delivery, technology people, synthesis of lipids, protein engineering, fusion protein, etc., molecular antibodies, and people that do the biology, which is always more sophisticated than the technology. So we are very much interested in RNA, which we believe that will be the next protein engineering phase. So the RNA area is getting more and more attention, and I think Heinrich, after me, I'm going to give him a spoil here a little bit. So for us, leukocytes and RNA is much more interesting, and all the challenges for creating RNA delivery carriers for leukocytes have been around for many years, or in general, all of those have been around, but for leukocytes, it's an extra challenge. It's very hard to penetrate them. In some cases, even viruses cannot penetrate. For example, memory B cells, very hard to penetrate, and we try to understand also a little bit of the biology behind us. So less than a month ago, Alnylam got an approval for patisiran, the first RNAi drug ever. And this is very interesting and boosting the field of RNA. And it's made with a lipid nanoparticle. So I think this is really an amazing time for the people that are in the field. We I'd like to tell you a little bit of what happened historically. So historically, we published a paper about 10 years ago, 11 years ago, in Nature Nano that is still considered the highest cited paper in the field of nanomedicine, and cancer nanomedicine in particular. And then we were a bit naive about the EPR effect and many other things and the opportunities, and many things happened from that point. But about a year ago, we were asked by the Nature Publishing Group to revisit this after 10 years, and recently we published this Progress and Challenges towards Targeted Delivery of Cancer Therapeutics. And if you have read this review, it's a bit of a depressing review. And the reason it's revisiting many things that we already know, but also putting it in perspective. And one of the things that we learn more is the bio-nano interaction from different areas, from the immune standpoint, from the field of basically of distribution, and the problem of specificity is still there. So we don't have really an active cellular targeting approaches, and this is what my focus here will be. So human diseases are heterogeneous, we all know about this, and even cancer has different phases. And inside the tumor, we have different types of cells. And inside these cells, we have different receptors. And to target all of them will be basically meaning we have to target them in different drugs. Why not to develop a platform that by a simple switch, you can potentially go after different subsets of cells? So we started thinking about the idea about 10 years ago, and we published this first paper, which became a seminal paper in Science. But then there are lots of challenges in order to get it into a scale-up process and a very easy process. So the challenges include chemical conjugation of your targeting moiety, and it's also include an example that antibodies are not alike. They have different glycosylation, so the chemistry is different. Actually, it's very poor in terms of globular surfaces. So if you want to conjugate any monoclonal antibody in any chemistry to globular surfaces, and you do it carefully with radioactive tracers, you will find that the efficiency in most cases is very poor, less than 5%. Although in the catalog of the company it says 70%. But that is true for planar surfaces, not for globular surfaces. And it's very tricky because you can never control your orientation. So we thought, and also, of course, there is an option of FC exposure. And luckily, immune cells and other cells have FC receptors, and they like to uptake things, especially if you have those nanoparticles. We have different antibodies, different orientation, so the clearance could be very, very fast. So we thought about how to do it a little bit differently, and we thought about going back to biology. So from chemistry back to biology, and thinking about affinity. And this is a bit tricky, because affinity has also some issues. But if you can create an antibody that recognizes basically the FC portion, basically a secondary antibody, and you can conjugate it to the surface of a particle, then you can control the orientation. By a simple switch of the same isotype, you can have a platform. And this is what I'm going to talk about this. And the first thing is, to avoid more nuclear phagocytes, unless this is your target, this is a schematic illustration of a particle. Just get rid of the ears here. Okay, so what do we do in order to avoid this? We pegylated this. And this is the pegylated version. I want to thank Moin Moghimi, always, for sharing those pictures. And also, it's better to orient your antibodies or your targeting moieties, right? So this concept, we conceived it a few years ago, together with two very talented graduate students, Ranit Kedmi, who is now a postdoc in Dan Littman's lab in NYU, and Nofar Vaiga, who is still looking for an interesting postdoc. And Ranit basically was the first to initiate this concept, and she really nurtures Nofar, really nurtures her. And you can see at the end how the student look at his mentor. Actually, I like those pictures. I took them a few months ago in Rwanda. So basically, creating a precision gene manipulation to subsets of leukocytes is very challenging. But again, using this platform, we thought that it should be a piece of cake. And together with Itai Ben-Ar, who is a protein engineer at Tel Aviv University in our same school, we basically started by generating 3 different antibodies, a mouse anti-rat IgG2, a mouse anti-human, and a human anti-human. Then we made a single chain based on them, and then we lipidated with 1 transcript. So we have a single chain lipidated. And the idea was to include this into a lipid nanoparticle. Using microfluidic system of precision nanosystem, I think they're outside. Basically, we took the RNA payload, the lipids that are ionizable lipids, and then creating the particle, then bacteria produce it, this lipidated single chain. You can actually get mice cells, and then again on the microfluidics, you can get a very robust system. And you can scale it up. We can scale it up to a liter at this point. It's very nice, very precise in terms of the ability to bind. And then this is a schematic illustration. And just to show you the versatility of this platform, I'm going to go quickly because I see I don't have any more time. Okay, everything, you can read it. This was published a few months ago. And basically, just to show you that you can go, after different directions, you can include, for example, a fluorescent marker, or not a fluorescent. In this case, it's a Cy5. You can go after subsets of leukocytes. This is an anti-beta-7 integrin that goes after all leukocyte subsets, anti-CD3 to T cells, anti-CD4 to subsets of T cells, anti-CD25 to subsets of CD4. And then injected to the tail vein of a mouse, and then 1 hour later, look basically what's going on. And again, I don't have time, but just to show you that it's very selective in vivo, and you can go after those subsets of cells. Moreover, you can actually do a selective knockdown approach with this. So here we use entrolizumab, an anti-beta-7 antibody in development with Genentech. And you can see actually that this antibody against the beta-7 targets both CD4 and CD8. And the hypothesis was if we use only a CD4 antibody, okay, it will go only to the CD4 cells, whereas the anti-beta-7 will go both to CD4 and CD8. And just to show you that it actually works, we encapsulated in this system an anti-CD45 siRNA, and then we put basically CD4 or the anti-beta-7 injected into mouse, and then 5 days later took the splenocytes, and you can see that if you look at the gated population of CD4 and CD8, and CD45 expression is knocked down with a beta-7, both in CD4 and CD8, where the CD4 only in the CD4 population. And does it worth anything therapeutically? And the answer is yes. We did 2 models. We used the colitis model, DSS colitis, and a mantle cell lymphoma model. Here, the target cells were inflammatory leukocytes that express the Ly6C population, receptor, and the target gene was TNF. And in any parameter, basically, so this is going after mononuclear phagocytes, and those inflammatory Ly6C high cells are going up in the DSS colitis model, mostly at the day of 2 to 3, and then 3 IV injection with those particles that encapsulate TNF-alpha, basically demonstrate therapeutic advantages. And in the mantle cell lymphoma, we use the model that we have developed and published a few years ago. There is a paper in PNAS in 2016 on the model. And here the receptor targets was beta-1 integrin, or the CD29. The target gene was porlikinase 1, just as a proof of concept. And what you can see is basically that you can prolong the survival by blocking the cell cycle. This is a G2M blockage. And it's not enough, because at the end they will all die, but this is significant. Okay, so the linker approach has a few advantages. One, it provides you 97% efficiency in terms of the conjugation. It gets a correct orientation. It's preserved the antibody activity. So this is a trick of protein engineering, but a very trivial trick. You need small amount of antibodies, about 250-fold less than any published paper. And there is no need to really do purification. No colon is needed at the end. Although we have compared it, there is minimal endotoxin contamination coming from a bacterial origin, originally. It's versatile. And you can, if you read the paper, you'll see that we shield it from Fc receptors. So in terms of looking at this, we think this is a good platform. And for us, we call it a universal. I want to fantasize here that we can take lipid nanoparticles that have different cargos and FDA-approved antibodies. In this side, we have about 15. In here, we have 71 monoclonal antibodies. And hopefully, just combine them together in an easy way. I know it's not going to be easy, but in the academia, we are allowed to fantasize. So just a few more slides at the end. Where are we going? If this is not your target cells, I think it's important to avoid them. High specificity and functionality. I didn't spend time on this, but kinetics is very, very important. It needs to be a very fast internalization, very fast release. Seconds, okay? So in human PBMCs, we see this under shear flow condition in 45 seconds. Very fast. And of course, we always need better RNA drugs that will not induce immune response per se, or in the context of the carrier. Last but not least, funding agency and the people that worked here on this project. And you can see this is something I'm advocating in the last 2 years. Do a group meeting once a week, early in the morning with a glass of wine. Why is good? Apparently, your student will be very, very creative at 8:30 in the morning. Red wine. Okay, and I'm also advocating because I'm part of a winery. So this is the only real reason in the Golan Heights. Thank you so much for listening.
Danny, that's a great demonstration how a method can change the life of a project.
Yes, definitely.
I think fantastic demonstration. And Danny's presentation is open to questions.
Somebody there.
Yeah, go ahead. Avi, go ahead.
Danny, great talk, truly inspiring. I did want to ask you, how many MABs do you want to have per particle? Is there approximately, is it size related? Is it weight related? How would you?
I think if they're in the right orientation, you don't need more than 3 or 4.
Okay.
So in the past, we explored this, and we really thought that 20 is a magic number. But actually, if the KD of the antibody is very low, that will create a major problem of binding barrier. So they will decorate the surface of the cells, but they will not internalize. And most of the antibodies are not internalizing antibodies. So you have to pick and do a lot of cell biology to pick the right that are internalized and the fast one. Now, in solid tumor, it's much more complex because they are not internalizing so quickly.
But that's part of the secret, Danny, what you said, and you didn't stress it enough. Fast internalization.
Fast internalization.
Otherwise,
It will never work.
You go against yourself.
Yes.
You get a good labeling, and that's all.
Yes.
Okay.
It's good for labeling.
Yeah, fantastic.
Yes.
We did it 20 years ago. Yeah, go ahead. More questions? Yeah.
Thank you very much for your great talk. I did some work on these systems in the past, other systems, and I know that I have a question to compare with release, because release is very important for RNA therapy. It's actually vital. You can get things inside, but if it doesn't release, it doesn't work. So the different types of antibody, does that affect release, or the release is always very good, depending on what kind of antibody do you use? Thank you.
Okay, this is a very good point. So, to make a long story short, I will tell you you have to select. You know, many receptors has many antibodies. Let's take one receptor as an example. You can find LFA1, alpha L beta 2 integrin, has about 65 monoclonal antibodies that you can get them commercially. Okay? From what we have done, and we have a very nice paper in 2007, another PNAS paper, that we have shown that only 2 are internalizing. And these are ligand-binding domain antibodies. So they're actually, if you take the crystal structure of LFA1 and ICAM1, they're the one that binds to the headpiece of the integrin. So you need a little bit of cell biology, not a little bit, a lot of cell biology to identify the right pair between the receptor and the ligand. And it needs to be, again, fast, very fast, not trivial.
Okay.
Thank you.
Daniel.